Hi, I'm Helen. I am the dietitian at Dnalife, and I offer a lot of the client support and education support for the genes included in our DNA diet, DNA health, and DNA estrogen tests. The reason for the presentation today is just to give you an introduction to the new genes that we have added to the DNA diet and the DNA health tests. And so that when you are going through these reports with your patients and you see these new genes, you are familiar with them and you are able to communicate the interventions to your clients more clearly. The reason for us updating our reports with new genes is that we are always trying to keep up with the science and the latest science that comes out. And we are constantly reviewing the literature for these genes. And as genes come up that fit our criteria for including them into our reports, then we will add them to the relevant tests. And so 3 genes have been added to the DNA diet test: the DRD2, SLC2A2, and TAS1R2. This is just a sample report to show you where they will be included and which area of activity they will be included. The main thing to remember with these 3 genes that have been added is that they are all very much involved in the carbohydrate sensitivity or carbohydrate responsiveness section of the DNA diet test. Starting with the D2 dopamine receptor gene, or DRD2, and I've just put here what the risk allele will be just to make it easier for you. Just to give a bit of background on this gene is that DRD2 is very much involved in the reward circuitry pathway. So midbrain dopamine circuits may play an important role in both addiction and normal eating behavior, as they are involved in reward processing, particularly dopaminergic signaling by the dopamine receptor 2 or DRD2. So this gene has been commonly linked to alcoholism as well as other addictive behaviors and overeating. And the TAC1A1 or T allele is what you will see in the report, as you'll see the T allele as the risk allele, has also been associated with obesity, comorbid substance use disorders, cravings, and reward seeking. And so consumption of sweet or high-fat foods, and often a combination of sweet and fatty foods, increases dopamine concentration. So eating chocolates and cookies and rich foods will have an effect on dopamine concentrations and activate this dopaminergic reward pathway of the brain. And in individuals who carry the T allele, there has been a greater consumption of sweet foods found. And thus with this T allele, these individuals, so the TC and the TT individuals, tend to have higher consumption of sweet foods and fatty foods, and also are associated with having an increased BMI. And so when you see the T allele coming up with this gene, it's important to note that these individuals may have tendencies to seek out sweet and high-fat foods, and then the intervention here would be to avoid all high-sugar foods and high-fat foods and really to reduce total carbohydrate intake, so to try and decrease that behavior associated with this gene. Secondly, we're moving on to a very interesting area, and then that's how we will introduce the next 2 genes involved in DNA diet. This is where we are looking at the genetics of taste. And it's very important to note here that you may not experience taste the same way as your patient experiences taste. You may find something very sweet where you have 1 teaspoon of sugar in your tea, and the same amount of sugar in your patient's tea they won't even taste it and they won't experience that same sweet sensation. So it's very important that people taste bitter differently, they taste sweet differently, they taste all sorts of different foods differently. And so when you are compiling a meal plan or giving recommendations, you must remember that your taste sensation will be very different to that of your patients. Something else to remember about taste is that it was generally thought that taste just existed on the tongue, that we had different taste bud regions, one area for bitter, another for salty, another for umami, sweet, sour, fatty, etc. And that's where taste stopped. With new research coming in, we have found that taste does not stop at the tongue. It actually is involved in a whole lot of other areas associated with the gastrointestinal tract. And so when we are looking at the next genes coming up, we will see that these genes are actually expressed in a whole lot of other tissues and not actually the tongue. So just starting with our first gene, just a bit of information here. Twin studies indicated genetics explained around 33% of the variance in intensity ratings for a suprathreshold sweet solution and 53% of the variance in frequency use of sweet foods. So that's quite a significant number, and that's very important to keep in mind. And taste is an important determinant of food consumption, and genetic variations in the sweet taste receptor subunit, TAS1R2, may contribute to these inter-individual variations in sugar consumption. And the diverse tissue distribution of TAS1R2 gene affects food intake beyond the detection of sweet taste on the tongue and palate. So as I said, these genes are expressed in a number of different tissues, including the gastrointestinal tract, the pancreas, the hypothalamus, and other tissues known for regulating metabolic and energy homeostasis. And that's very important to take note of, is that taste is a very important determinant of food consumption, and it is very genetically based. So moving on to this first gene that we've been speaking about now is involved in taste is the TAS1R2 gene, the taste receptor type 1, member 2. Here, the risk allele is the A allele. And what they found is that compared with individuals homozygous for the GG genotype, there is a lower consumption of sugars. Compared with individuals homozygous for the AA genotype, there is a lower consumption of sugars in the G genotype. So here we are seeing that AA and GA individuals will take in a lot more sweet foods and seek out sweet foods, and that there is that association with that high consumption of these sweet foods and a higher obesity risk. And so again, in this area, it is very important to remember that we must decrease total intake of highly refined carbohydrates and put in a whole lot of behavior interventions that will decrease that individual seeking for sweet foods. So again, decreasing in total carbohydrates and sweet foods will have a positive effect on weight in these individuals. Moving on to our next gene, which is involved in taste sensitivity, is the glucose transporter type 2, or the GLUT2, and this is coded by the SLC2A2 gene. So in the report, you will see that the SLC2A2 gene is mentioned, and in this gene and the SNP, the risk allele is the T allele. So just a bit of background again here. GLUT2, coded by the SLC2A2 gene, is a member of the facilitated glucose transport protein family, or the GLUT family, and is expressed in the pancreas, the liver, the small intestine, kidney, and brain. And so again, just to point out that taste does not stop on the tongue. GLUT2 facilitates the first step in glucose-induced insulin secretion with the entry of glucose into the pancreatic beta cell. And because of its low affinity for glucose, it has been suggested as a glucose sensor and is considered to be important in the postprandial state. It is also involved in food intake and regulation. So the GLUT2 null mice, and this is where what happens is it's a study where they take a gene out of these little mice and then they see what happens to them. They can also be called knockout mice. So in GLUT2 null mice, these mice failed to control their food intake in response to glucose, thus suggesting a potential role for this transporter as a glucose sensor in the brain. And because GLUT2 facilitates the first step in glucose-induced insulin secretion, with the entry of glucose into the pancreatic beta cell, it's also very much involved and very important in one's response to glucose intake and sweet foods. So this gene has also been associated with risk of type 2 diabetes, and where the TT and CT genotype, or ill carriers of the SNP, consumed more sugars from sweets such as baked goods and chocolate and sweetened beverages. So specifically, you could almost call it the junk foods. They had a real seeking out for these foods rather than trying to take in more fruit. And the reason for this has been attributed more towards glucose sensing compared to glucose insulin levels, and this is what affects these individuals' food intake. So again, intervention in this case for the CT and TT individuals is to decrease total carbohydrate and especially refined carbohydrate intake, and then that may decrease or show better results for weight management. Okay, so those are the new genes in our DNA diet test. Moving on to DNA health, we have also included a whole new panel, which is our food responsiveness panel, and then we've also included the SLC2A2, which I just spoke about, to the insulin sensitivity because it is involved in or associated with risk of type 2 diabetes. And then I just also want to mention something about PPAR gamma and the new impacts that we have put there. So again, just a little recap on the glucose transporter type 2 gene, or the SLC2A2, which encodes glucose transporter type 2. This is an integral plasma membrane glycoprotein of the liver, islet beta cells in the pancreas, the intestine and kidney epithelium. It facilitates, as I said, the first step in glucose-induced insulin secretion with the entry of glucose into the pancreatic beta cell. And as you can see, there is a strong relationship here with type 2 diabetes, where you have the CT and TT variant because of this entry of glucose into these pancreatic beta cells. And so with the variant, the risk of type 2 diabetes does increase. And so where you see the variant, it is important to stress decreasing total carbohydrate intake, specifically around your high GI and highly refined carbohydrates. Okay, so that's the gene that we have added now to the insulin sensitivity panel. And now just to move on to PPAR gamma, which I'm sure you're all familiar with. It's been the most researched, a very well researched gene, and has been the longest standing member, if you want to call it that, of our insulin sensitivity panel. It's also included in the DNA diet test because it has a significant role in weight management. So PPAR gamma is significant in both lipid and glucose metabolism, and the SNP is well established in research. But with more genes being added to the insulin sensitivity panel, we have re-looked the impact factor for the PPAR gamma genotypes. And so, because the DNA health test has evolved, and because the DNA health and the insulin sensitivity panel has a number of extra genes from when we started with the test, these genotypes do need to be evaluated in relation to the SNPs in the metabolic area of insulin resistance. And so once we evaluated this, and we considered the relationship of all of the genes, we have decided that the PPAR gamma CC genotype will now have a medium impact, and the CG and GG genotypes will have a low or no impact. And so it's still a very important gene to consider, but just to consider it in relation to the other genes and SNPs in the panel. So that's just what we wanted to mention around PPAR gamma. Okay, now moving on to food responsiveness, we have a number of new genes added to this panel. It's actually a whole new panel in itself. That's a very exciting and very interesting panel, I think, especially for your patients. So starting off, just to introduce these new genes, we have the FADS1 gene involved in polyunsaturated fatty acid metabolism. We have the CYP1A2 gene involved in caffeine metabolism. MCN6 involved in looking at adult lactose intolerance, and then we are looking at ACE and AGT in salt sensitivity and blood pressure. Okay, so let's start with PUFA metabolism and looking at the role of polyunsaturated fatty acid metabolism in an individual's health. And so tissue availability of polyunsaturated fatty acids depends on dietary intake and metabolic turnover. It's very important to note that optimal levels and ratios of your different polyunsaturated fatty acids have a major impact on human health, as it influences or mediates inflammation. It supports infant growth and brain and neural development, as well as having a very important role to play in immune function. And so this is a very interesting section that we have added to this section. So just to go through this pathway in a little bit more detail, the delta 5 and delta 60 saturases, so you can see over there, delta 5 and delta 6, encoded by FADS1 and FADS2 genes, are key enzymes in polyunsaturated fatty acid metabolism. And they catalyze the conversion of your linoleic acid into your eventually arachidonic acid, as well as your linolenic acid into your eicosapentaenoic or your EPA, and your docosapentaenoic acid, hexanoic acid, your DHA. And so SNPs in the FADS locus have been associated with blood concentrations of long-chain polyunsaturated fatty acids, as well as with cholesterol concentrations and mediating inflammation. So based on genetic variation in these genes, individuals may require different amounts of dietary polyunsaturated fatty acids or long-chain polyunsaturated fatty acids, so these bottom ones here, to achieve comparable biological effects. And so this is a very important area to understand, and the gene that we're looking at in this area is the fatty acid desaturase 1 or FADS1, and it's a G to T chain nucleotide change, with the G allele being the risk allele. And so just to show you again, variations in this FADS1 gene cluster are strongly associated with fatty acid composition in phospholipids. And the enzyme encoded by fatty acid desaturase 1 is a rate-limiting step in the desaturation of your linoleic acid to arachidonic acid, as well as your alpha-linolenic acid to eicosapentaenoic, which is your EPA, and your docosahexaenoic acid, which is your DHA. So it's converting those fatty acids to your longer chain fatty acids. And so with the G allele, we find that there is an increased conversion, or there's an association with an increased or enhanced conversion to your arachidonic acid due to increased enzymatic efficiency of this pathway and of this enzyme. And thus there appears to be associated high levels of your arachidonic acid, which, as you would know, is a pro-inflammatory fatty acid. And so then it has also been associated with systemic inflammation and inflammatory disorders. And so in the presence of the G allele, it is important to decrease pro-inflammatory fatty acid intake, which is generally found in the modern Western diet. So really to focus on that individual's diet in terms of the fat composition, and then also to ensure adequate intake of your active EPA and DHA fatty acids. Moving on to caffeine metabolism, here we are looking at a phase 1 enzyme, and the gene that codes for that is your cytochrome P450-1A2, your CYP1A2, and that is an A to C nucleotide change, with the risk allele here being the C allele. So just a bit of background, this gene has often been referred to as the coffee gene because coffee is such a major source of caffeine, and the main gene that metabolizes caffeine in the body is your cytochrome P450-1A2, which encodes for the enzyme, your CYP1A2. So what they have found is that individuals who are homozygous have 2 copies of the A allele are rapid caffeine metabolizers. This means that they metabolize caffeine very well. In carriers of the variant C allele, so AC and CC genotype, these individuals will be slow caffeine metabolizers. And so these individuals will not metabolize caffeine as well. And it is important to note here that just besides being awake at night because you're not metabolizing as fast, is that individuals who are actually taking in caffeinated beverages and who have the C allele, so the AC or CC genotype, are actually at increased risk of cardiovascular disease or heart attacks when there is a high intake of caffeine and when they are a slow caffeine metabolizer. So in these individuals, when you see the C allele come up, it is important to decrease or actually try to cut out caffeine intake in the diet. And that includes your coffee as well as other caffeinated beverages such as a lot of your energy drinks. Okay, Moving on to lactose intolerance. This is coded by the MCM6 gene actually. So MCM6 gene helps control the expression of the nearby gene LCT. And LCT encodes the enzyme lactase. So a bit of background around this. Lactase is the enzyme which is found in your GIT, which helps to break down lactose, the sugar found in milk. If you do not have lactase or you have insufficient amounts of this enzyme, lactose cannot be broken down, and thus you will experience those uncomfortable side effects of lactose intolerance, being bloating, gastrointestinal upset, etc., etc. So where there is the C allele present, which is the risk allele, the CC genotype specifically, these individuals have been found to have decreased lactase production and thus decreased ability to digest lactose, specifically in adulthood. And this gene was really found to be the gene that we look at for adult lactose intolerance in the Caucasian population. That's just something to note, that this is different there is a different gene that gives the same information in the African population. So in the Caucasian population, the risk allele C for MCM6 will give you increased risk for lactose intolerance in adulthood. So, of course, it's quite an obvious one. Intervention would be when you see the C allele, decrease or avoid lactose and lactose-containing products. Okay, moving on to salt sensitivity. This is where we are looking at one's ability or one's risk for increased blood pressure with higher salt intake. Here we are looking at two genes, starting with ACE. This you will see also in the DNA sport test. ACE is also very much involved in power and endurance activities, but here we are talking about it specifically in terms of risk for blood pressure, higher blood pressure. So here the risk allele is the I, or insertion, and where ACE codes for the angiotensin converting enzyme and is part of the renin-angiotensin system. This controls blood pressure by regulating the volume of fluids in the body. And studies show that patients with essential hypertension, homozygous for the insertion allele, in other words, who have an II genotype, have a significantly higher blood pressure increase when there is a high salt intake compared to their DD counterparts. So just to note, if you see the ID or specifically the II genotype with the ACE SNP, you must really focus on decreasing sodium and total salt intake in the diet to avoid these blood pressure increases. The second one that we're looking at in this panel in salt sensitivity is AGT, which stands for your angiotensinogen. And this is expressed in tissues involved in blood pressure regulation, such as the kidneys, adrenals, and the brain. So again, it's involved in blood pressure regulation. You also will find this gene in the DNA sport test, but again, we are speaking about it here in terms of blood pressure and salt sensitivity. So increased angiotensinogen levels correlate with increased blood pressure. And this gene also influences salt sensitivity of blood pressure, where individuals who carry the C allele, so here the C allele is the risk allele, and so if you see the CT or CC genotype, this is where you should note that individual's salt intake and decrease it, because in these individuals there is the incidence of hypertension was found to be significantly lower among those individuals who reduced their sodium intake. So decreasing sodium and salt intake will decrease risk for hypertension.